Okay, so I'm happy to welcome everybody to part one of our workshop series. That is a collaborative effort between GrasPods and the BC Children's Hospital Research Institute's trainee Omics group, or TOG. So this workshop today is part of the extended edition of our GrasPods Welcome Week that is extended into October, and will give us an introduction on how to go from raw to processed data in preparation for a downstream RNA-Seq analysis. So it's my pleasure to introduce today's speaker, Nikita, who is a PhD candidate in medical genetics at UBC. She's currently co-supervised by Dr. Juan Lam at the BC Cancer Research Center and Dr. Wendy Robinson at Children's Hospital. And her research focuses on genetic regulation during human development. She holds a bachelor's in biomedical genetics from Newcastle University and a master's in genetics of human disease from University College London. When she's not working on her analysis, she does enjoy reading, photography, and expanding her repertoire of cooking recipes and drinking Italian roast coffee. So with that, I'll transfer it over to Nikita.
Hi, everyone. Thank you so much for the introduction, Lorenzo. I think you did a great job about the small tidbits. But yes, as Lorenzo mentioned, I am a now third-year PhD candidate at BC Children's and at BC Cancer as well. And my focus, the focus of my research is primarily characterizing novel and known small non-coding species in the human placenta. And for this, we are doing some very small RNA-specific RNA sequencing, and that's how basically I got into RNA-Seq data analysis. This is the second time that we're running this workshop. So given that I'm part of the trainee Omics group, I'd like to just introduce who we are and what we do. I have our brilliant TA, Christina, with me over here, and she'll be answering any and all questions that you might have. I will also be sharing this link in the chat now. So this is where if there's a big chunk of code that just needs to be copy pasted because it takes a little bit more time to write, the code will be posted here. And if you have any questions, just use the pound or the hash key, put in your question, and Christina will be answering that. Okay, so an introduction to TOG. Let me just bring that up. Okay, can everyone see this? Okay, perfect. So the Trainee Omics Group is an organization based at the BC Children's Hospital, and we are a group of trainees from both dry as well as wet lab projects with a common focus to bring about support for data analysis and mostly bioinformatics. So we focus mostly on biomedical research. So all the statistical analyses and methodologies that are involved in health research, we kind of go over those. And the workshops that we have, or like all of the events and resources we have, are mostly open to trainees at BC Children's, but certain events such as this one are open UBC-wide. So just a brief overview of what we do offer is that we have some short tutorials with the topics mentioned over here monthly. And these are mostly very focused on very small topics, which are part of, a bigger research analysis. We also have monthly seminar series where it's either one faculty member who talks, who has knowledge about big data, who presents, or two trainees who come and present their work in a more of a feedback manner rather than a full-blown presentation that you might have at a seminar. So this really helps with the discussion that you might have in regard to a project where you might want to take it further, and it's quite low pressure as well. Workshops such as this one are usually longer, and they are geared towards more of the requested topics by our community. So this RNA-Seq workshop was quite highly requested. We also have an intro to our workshop, which is happening in a couple of weeks. And then lastly, we also have drop-in sessions, which are going to be starting bi-weekly now. And these are the sessions where you can bring in specific problems that you might have, okay, I have a certain threshold of blah, 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 but what if I use this threshold? So those kinds of, conversations which are one-on-one. You can just sign up and drop in, and one of our committee members or our volunteers will be there. And this is our main trainee Omics group. exactly like Christina's over here, we have volunteers as well. So if you do want to join, just email us. And we have a position open for digital director, but you can read about that later. And these are all the ways to get in touch with us. So our main GitHub page, our website, our Slack, and our Twitter. And then if you want to sign up to a mailing list to know all of the events that we're going to be putting out for this year, as well as our Slack group, you can just scan this QR code on your phone, and then you'll directly be signed up. And so that is for TOG. And now I'll basically start a little bit about talking about the workshop. So we will be running through raw RNA-Seq data and then getting it to a processed format. So this format is basically what you use to perform differential analysis, network analysis, but there's a lot of pre-processing steps that happen before that. And so I'll just share at the end of the tutorial what you would get as the output. Okay, so this everything that I'm going to be talking about today is available on our GitHub page over here. So if you go into BCCHR trainee Omics group study group, RNA-Seq workshop, you have all of the files that you would require. And even if you do missteps in the tutorial, each of these files, which are basically labeled as TOG RNA-Seq workshop part 1 or 2, has a full write-up of all the steps that we will be performing. So I will be going slowly, but this resource is still there for you if you want to do it on your own in your own time. And yeah, so at the end, this is something that you're gonna get, and we're gonna go step by step to make all of these plots and do all of this analysis. And as in the introduction, we will be going through a couple of steps as pre-processing, filtering, and normalization, and ending with saving our data, and which we will be able to use in part 2 of the workshop, which I think is happening sometime in October, but I'm sure GrasPods will let you know about that. Okay. So in the email, I hope that everyone has actually completed the pre-workshop to-dos. Can you raise your hand if you have not? Okay, there's a couple of people here. Okay, so I'll go through what we need to do because we will be needing some of these files. So if you do go, Christina, would you mind posting the link to this in the chat? So if you do go through this document, you'll see that as long as you have R and R0, please don't update that right now because that does take a little bit of time. You will have to install these packages. So if you do put in exactly this, just like copy paste it into your R, it should install the packages that we need. You need to create a new project, which I can show you how to do in just a minute. But the biggest thing is that you need to download these 3 objects over here. I can show you how to do that. So if you go to our main page over here, and you go into data, you'll see these objects over here. The Pdot, which stands for phenotype data frame, EDAT, which stands for expression data frame, and a BAM-R object, which is a R object. And if you want to download that, you just click on that. It won't be able to show you anything because it's too big of a file. You can click on download, and it should give you something like that. Now, you have to let it finish loading over here. This might take a little bit of time. Yeah, okay, it's done downloading. And make sure that you're not selecting anything, so otherwise it won't work. So just over here, right click, and then save as, and you should get a text box just saying to save it. So just save it as a .txt file in the data folder that you'll have, because the formatting and the hierarchy of this folders is quite important. The same way to do with the other EDAT, like both the EDAT and PDAT files, and then the object, you do the same thing. So download, and then right click. Make sure that the extension is .rds for the BAM file, BAM R object file, because if it's text, then it won't open properly. And lastly, I did upload a worksheet in the scripts folder, which contains a little bit of code, which is just for graphing, but it's just easier to have there instead of copy pasting. So if you just go over here, the worksheet, and you go to raw, again, right click. Oh, no, that's not right. Right click, save as, and then part 1, and make sure that the extension is .rmd. Do remove the txt at the end, and then save that in your scripts folder. Okay, this is where we are. I'll pause for like 2 minutes so that everyone can get up to speed. Okay, until everyone is doing that, I'll just show how to make the R project for those who are new over here. Okay, so once you open R, you'll see something like that, loading your R version, whatever that is. This is not the latest version, I know. But again, just as long as it's working, it's fine. And then what you do is go over here, this blue icon here saying create a project. Okay. Right, okay. I'm hoping that you can see this create project window. Christina, can you see the window?
Yes, we can see it.
Okay, perfect. So then if you just click new directory and then new project, and you just browse and like go wherever you want to put your project in that directory, and then just the name. So I've named mine TOG RNA-Seq 2021. And then within that, I will just open mine because I already do have the project. So open project. And so then when I come over here, I'll see that if I go here, these are the folders that I have: data, scripts, static. And so the data folder contains all of your data files. Your scripts are all your R files. Static is usually reserved for some images that you want to put in. So we won't, like, that's not really necessary. But as long as you have the data and the scripts folder, with the data folder containing 1, 2, and these 3 files, we're ready to go. And then if you go into the scripts folder, and I know there's a bunch of these here, but then just the worksheet one, we just open that and we're ready. Okay, so if someone is still stuck or still doing these steps, can you just raise your hand so that I know to wait? Okay, looks like everyone's up to speed. Perfect. Okay, so let's start. Okay, so now that you have your worksheet open, just as like a disclaimer at the start, I really advise everyone to be as vocal as possible, because the way that you learn when it comes to all of these things is the more that you talk, the more that you're gonna, figure out your question. So just if you have a burning question that you think everyone in the audience might like to hear, just speak up at any point. Okay, now how many of you actually have, can we have a poll? Okay, you know what, I'll just go through this. So when you start with RNA-Seq analysis, the first thing that you get is a FASTQ file. Now, does anyone know what a FASTQ file is and what it contains? Just speak up, it's totally okay if that's not right. Okay, so a FASTQ file is basically the raw sequencing file that you get from the sequencing center. And this contains all of your reads. However, these reads are not aligned. So, you know, you have all of the reads that have been sequenced using your primers, your index adapters, and all of that. Now, once you get your FASTQ file, there are a couple of steps that you might have to go through, you know, trimming the adapters, making sure that it's only the sequence that you like, you want to only retain the sequences that you have. So just as an overview, I would say that for my files, I would have to trim the UMI, which is the unique molecular identifier at the start. Then I have my microRNA sequence, I have my index adapter, I have my index primer, I have my adapter. All of these things I need to remove. And then I need to further trim down to only the sequences that are about 17 base pair in length, because that's the minimum length of microRNA. So depending upon what type of RNA data that is, there are a few pre-processing steps that you will have to perform. However, given that a FASTQ file is a couple of gigabytes, we will not be going through that. However, I can, if you do go into the part 1MD file in the scripts, you can see an image that actually displays what the data in a FASTQ file looks like. There's 4 lines, it tells you the sequence, the quality, the score encoding, and all of that. And that FASTQ file, then because it's an unaligned file, you need to then align it to the reference like the human genome, right? Now there's versions of the human genome, and for those, you specifically require a couple of steps that you need to perform. So I'll stop sharing my R here, and I'll show you what it looks like in our study group. So there is a file called BAM-FASTQ to okay, so there is this file which you can find in workshops, RNA-Seq workshop, data, and in just this file over here. And this one, you can download it and you can use it. This just goes through the basic steps, how to convert unaligned FASTQ files to aligned BAM files. Now, you can either get BAM files, which is a which is an equivalent of something called as a SAM file. Now, a SAM file is something, a file that is human readable. So if you open the file, it's a text file, and you can see everything like the sequences, the name, and everything that the FASTQ file contains once it's aligned. However, computationally, a BAM file is used because that SAM file, if converted to a BAM file, which basically just means binary alignment mapping file, it's much easier for the computer to process rather than the SAM file. So usually you convert your FASTQ into a BAM file. And if you just go through these steps, which takes a little bit of time, but I've tried to annotate this as much as possible and then shown exactly, you know, which flag means what. That's basically how you would convert your FASTQ file to your BAM file. And you have to do that for each of your samples. So each sample will have a separate FASTQ file. You can also convert your BAM files back into FASTQ. So if you want to align them to, you know, right now the current version of the human genome is 8G38. But if you want to align it to 8G19, which is the previous version, you can convert your BAM file back to FASTQ, and then align it using 1819 and then convert that to a BAM file. So everything that I've used and is like the most basic level, I've put in this PDF over here. So just feel free to go and download it from here. And what we're going to start off in the workshop is the directly the BAM file. Now, for those of you who did the pre-workshop to-dos, you would see that if you go into your data folder, you would have this file, which is a BAM file. It's from the Human Genome Project phase 3, so the latest version, and it is a female sample of European origin. Now,
Nikita?
Yes, sorry. Am I going too fast?
No, there's just a question in the chat. Someone's asking if trimming is necessary if you use soft clipping with aligners such as star. Do you want to comment on that?
Yes. So what I would say is that it's much easier to get all of your trimming done before you put it into any alignment software. Personally, I found that if you just put in the sequences that are ready to align, that performs much better than having to trim or do all of these extra functions in star, which is the aligner that I use, and it's quite good for small RNA data. But you can use bow tie, you can use top hat. But yeah, I have found that if you trim it before, it performs much better, and it also takes up less time because ASR or like the program that you're using is not performing as many functions. Does that answer the question?
I hope so. If not, whoever had the question, you can just feel free to. Oh yeah, yes, thanks.
Awesome. Okay, anyone, I'll stop here before we start our coding. Any questions so far? Please speak up. Okay, perfect. So when you open your worksheet file.
Sorry, I have a question.
Yes.
So you don't have to, it looks like you don't have to code to align and everything, right? To the genome reference. So now it's done everything, like when you call that, it's all done by that feature option, feature counts.
Yes, so if you do have a BAM file, which is already aligned, this is the command that we'll be using. But to produce the BAM files, those were the steps that I was going through, like from FASTQ to BAM.
Okay.
Is that clear, or should I explain more?
Do you mind like explaining like quickly again? Maybe I missed that. Like, how do you go from the FASTQ
Sure.
like to
okay.
BAM? Like, it's not an hour, right? You showed something different.
No, it's not. Okay, sure. I will go through that. Let me stop sharing. Okay. So once you get your FASTQ file, so usually from any sequencing center, you get the raw FASTQ, but they also have the option to give you a BAM file, which is already aligned. However, because it's done by the sequencing center, they might have used some parameters that you might not want to necessarily use. So when I get my files, they're aligned using bow tie. So that's the BAM file that I get. No, sorry, Burrow-Wheeler alignment. Now that's not the one that I want to use. So that's why I want to convert my own FASTQ files. But if you're okay with what the sequencing center has done, you can like just go ahead and use the BAM files that you get. Okay, so then all of this is much easier done in a Linux or a Mac operating system environment. The first step is to install a package or software called as Conda via these two links. You can do it on Mac or Windows as well, but Windows is a little bit difficult when it comes to like these kinds of things that you want to run. So if you do have access to Mac or Linux, this is much easier. So then these are the steps that you go through. So you need to do something, you need to make something called as an environment to work in. So that's like a virtual machine that's running on your computer. And once you do that, you activate it. And within this, because we need to use a package called Trimomatic, which is part of the Conda software. So you need to install Conda before you can install this Trimomatic. Now, Trimomatic, Cutadapt, these are just the ones that I use. There are a few out there, but each one performs something a bit different. So you might find that one facilitates your needs better. And then you can just go to the website over here and then put in whichever parameters that you want. So for me, like I said, I need to remove the first 10 base pairs, which contains my UMI and a dinucleotide. So accordingly, once I go here, I will have a, if you go to the actual documentation here, you'll be able to see all the parameters that are offered. And then you can also do it by Cutadapt. So Trimomatic doesn't allow me to trim the index primer and the adapter sequence that I have at the end of my sequence for whatever reason. So I like to do that by Cutadapt. So I use both. Then this is just installing all of the software you'll require. Star is the aligner. Again, you use Conda to install all of these. And then this is an important step. You have to download the latest, or not the latest, or the version of the human genome that you want to use. It's usually present as a FASTA or a FA file. .fa is just the earlier or older extension of the FASTA. So it's the same format. And then the genome annotation file, which contains the location, the number of base pairs, and all of the meta information that you would require for the alignment. So these are the two files that you require in order to align your FASTQ files. You have to make sure that you download both of these files from the same platform. So I like to use ENCODE for that, but you can download it from UCSC, you can download it from NCBI, but you have to make sure that you download both of them from the same platform, because there might be a little bit of a difference between the two. Trust me, I've done that. When I use different files, it doesn't work. Okay, and then this is just a little computational thing like available threads. So how quickly can your job or like your command be processed? And then what you do is once you download your genome file, because it's a huge file. It's 3.2 billion bases. So it's going to be tons of gigabytes. And when you're aligning, especially short sequences, it's going to take a lot of time for the software to find that exact sequence throughout the genome, right? Because it's such a big file. So what you do is called as indexing the genome. So you divide the genome into small sections, which are just called indexes. And then that way, when you actually run the alignment software, Star takes in each index one by one and tries to see where it can be aligned. So it makes the job much computationally easier rather than it running for just, a really long time. So then you can use Star tool itself, make the index. That's the general commands that you would use. Each of these dash are called flags. So each flag has a value that you can give it. So run thread n means like number of threads to use, 10. Run mode, genome generate. So I've tried to write down all of the descriptions here. And then once you have aligned these files, you can either trim them down or you can do the trimming of these files before as well. It really depends what you want to do. And so that's basically here. And then at the end, you arrived at your trimmed FASTQ files, which can then be converted into BAM files by this little command over here. Zakir, was that a little bit better?
Yes, thank you.
Okay, perfect. Any questions so far on this segment? Okay, let's move on to our analysis. Okay, so we have opened up our worksheet, and this bit over here is basically you specifying the different parameters for a report that you'll produce. So an HTML, I want an HTML file. So that's what I'm saying over here. You can just leave this as it is. You can change this title, your author, date, all of that, like that's up to you. And then the theme and highlights is just something for the formatting. Okay, so when you start R, and I'm assuming there's a few R beginners here, in R, you work with certain packages to perform certain functions. Now, each function can be done in a different kind of way. So you can use 2 different commands or more than that for the same exact output that you want. We will be working through a language called as the tidyverse language, which is a bit more intuitive than the base R language, which is the, the basic level R programming one that you see. So the way to do that, as I mentioned, is through different packages. So if you have never used R before and you want to install a package, you simply do that by typing in install.packages and then the name of the package, whichever you want. However, I won't be running that. You can, if you don't have the package installed. So if you go back to the to-dos and then copy paste that, it'll install the packages that are needed. And so, yeah, I won't install this. Instead, if you want to load a package, you, so you type in library and the name of the package. And okay, so a few shortcuts over here is that when you write something and you want to run run it, you just click Command Enter or Control Enter, depending on which machine you are. This little bit over here is called as a code chunk in R. So all of your code is written in chunks. You can name your chunk after putting in the R, so that specifies it's an R chunk with these little parameters here. So let's say I've put in warning false, error false, message false. So I don't want any, like all of this output that I see over here, I don't want it to appear in my main file. So that's why I've put in message false. You can take that out. And then if you run this again, okay, it doesn't have any messages, but you will get quite a lot of output, which I don't want. So we'll just go through loading all of the ones that we need. Yeah, and then like it basically comes up with whichever ones you want. So let's say R Markdown, just run that. But I will copy paste that in the code share so that it's easier. So these are the packages that we will be needing. And okay, if you just copy paste that from the code share link. And so I specified these ones over here. So these are like the tidyverse package that I mentioned. Here is used for navigating through your directories. R Markdown is to produce the R Markdown document that will have, like this document that we're working in is an R Markdown document. And knitr will be used to make this into an HTML or a PDF or a Word file. And then these are just like extra packages that we require for, nice plots and stuff like that. And then if you run this, it'll run the whole thing over here. And then you can see, so here, which is the formatting, the directory package that we had, it says that it starts at this place over here. So that means that it's much easier to load in files if we go to our main folder here. And I'll show that how in just a minute. Okay, so everyone good till now? All our packages are loaded. And now we'll go into running, converting our BAM file into a counts file or an expression data set. Now this, again, like I said, this is just an example. You will have to do this for every single sample. So I've just taken this human genomes project sample and I'm just showing this to you. But the expression data set that we'll be using is one that I've already formatted from a paper. And it looked at COVID status between a couple of patients. So we're just going to explore that data set a little bit more. So that data set is the geo data set. I can't find the annotation. Oh, yes. So I'll just post that in the chat. So if you go to NCBIGO and put in this, you will get the data that I'm talking about. However, I have formatted that for the process of this workshop, so it's easier for everyone to work with because there's a little bit of pre-processing going on over there as well. Okay, so for those who did actually download the BAM file and make sure that it's in the data folder, if you use this command here, so align is the name of the object that we're giving. So this object will hold the output of the command that we're running now, which is the feature counts command. And actually, I forgot to load that. So it's from this package, R subread. You need to load that before you can use feature counts here. And this is where the here package comes into play. So like I said, here starts at TOG RNA-Seq Workshop 2021. And what I'm going to do is just specify the data folder. So it'll go into the data folder and then pull out this file here, which is our BAM file. And this is much easier to do than the base R setwd or getwd command, because that is not reproducible. When you're sharing your code, you have to make sure that anyone who is going to be using this code is able to reproduce the results that you want. So if I had put in, it's present on my Z drive and folder Nikita in GitHub, you won't be able to load that because that's on my local computer. So here helps you with making sure that your data is reproducible. These little 2 dots over here means that I'm going to use the here command from the here package, because there are 2 here. If I put in here, see, there's, if I just put in that, okay, it works here, but then usually there's 2 commands which have the name here, which is present in a different package. So sometimes you have to specify which command from which package. You put the double colon here, and then it knows that it's from the here package that I'm using this. And then I'm going to put in the 8G19 genome, because that's the one that they use to align their files. So you have to make sure when you're getting data from somewhere, what is the genome build? Like which reference genome have they used? Is it paired end? It is. So if you go and read through the documentation, their data is paired end. So we'll just specify these parameters and then run that here. And that might take a little while, but this is the output that you'll get. It's okay if you don't have the file loaded. I'm just showing you what it looks like. And it'll just give you a few statistics here. Successfully aligned 84%. And then it took like this many minutes, and then just like you can read about this in your own time. However, this is a pretty big file. So the RDS object, so if you now look at this, if you go over here and click on align, this is the output that you get. So the feature counts command outputs this object here. But what I did is also to reduce size, I actually saved this aligned as it is and a BAM object. Sorry, ARDS, like a R object. So for those who downloaded this one, if you run this command, let's call this align2 so we can see the difference. Align2 is basically the same. So what I did is just convert it into a R object. So this is something to remember when you're working with very big data sets, you can't actually always load in the Excel file because it might take time and it does take up a lot of space in memory. So if you just convert it into an R object, it's much easier to load in. R understands the format, everything is kept the same, is just present as a R object. And yeah, so that's what we get. And now if you, let's say, just go into align2, right? Everyone will have this. And you can just open all of these things and see. And now this bit that counts what can I call this? Like sub object of the align2 object is the actual counts that you get. So I want to pull that out. So I'm going to name my object expression counts. I'm going to use the aligned data frame. So the dollar indicates any one of the the subset of the file that data frame that you want to pull out. So if I just did that and then just put in $, these are all the different measurements that exist in the aligned data frame. So I just want to pull out counts, and I do that. I go over here, and there you go. You get your counts for your BAM file. Now these are entree IDs. This is something that will be required later on because, you know, sometimes people align it to the AGNC genome symbol, and then sometimes it's to the entree IDs, but you need to like figure that out as you go. But this is just one example of all the samples that you'll have to do. And now we'll start with our actual data. So okay, I'll just pause here. Any questions?
So, sorry, quick question. Will you have like missing data in the counts file, or is it like the output is always clean data so that it can work, or you have to do cleaning before converting to BAM?
Okay, so all the FASTQ trimming things that I was talking about, that is the pre-processing. So that is what you're doing. So if, let's say, like missing counts is in, I don't know which gene or transcript this corresponds to, this little ID over here, but it says 0 counts. That means that parsing through the FASTQ file, there were no reads that were aligned for this particular gene over here. So that's why it'll give you 0. And it doesn't mean missing data. It just means that it just might not be present. Yeah. Okay, I'll move on. Okay, yeah, cool. So then let's do this. So now in loading data, we are actually going to use the data that are formatted, and we can start with our analysis. So what I'm going to do is I'm going to name it Pdot. So that means expression, like E for expression, D for dat. That's just how I've been taught to do it. And then this command, read.delim, which means it's reading in a delimited file. And then here, specifying the package here. And then, like I said before, the data folder. And then usually if you just type in GSC and then tab, it'll give you all of the folders, but all the files present. You can go through this. Okay, I don't see it. Sometimes it works, sometimes it doesn't. So I'm just gonna add in the name of the file, this one over here, GSC 1517 103 formatted, no matches. Okay. Pdot.txt. And then because it's a delimited file, we have to specify what the delimiter is. So separation is going to be /t. That means tab. So that's a tab separation. Oh, actually, not this one, this one. Okay. Why didn't that work? So this is something that you're gonna like deal with quite a bit. Like you have to go through quite a few steps. Sometimes it doesn't work. So you can also do this. If you just, oh, the file is too large. Sometimes you can directly load it in. We did load the here package. So read.delim here data GSC 157. Yeah, there we go. So this happens all the time. Even if you have like a full workflow, you're gonna go through these things where sometimes something just, you've missed it. You miss a comma here or there, it's gonna not work. Okay, so we have our expression file. Great. So the first column is all of our genes, and all of the columns are our samples. So we have, if you look at it, 127 columns minus the first column. So that's 126 total samples. A very easy way to get, a little bit information is to type in dim, that is dimensions, and then just the name of the object. So that gives you the number of rows, that is 19,472. That's the number of genes, and then 127 columns. But like I said, the first column is for the genes. Or you can also do str. And then that actually gives you what type of column or what type of data do you have in your data frame. So the first column, gene, is it contains characters. So there's different classes of objects that you can have in R. So this first, it just specifying that this is a character. The second column is a numeric column, which is exactly what we want because these are numbers. But then sometimes you need to like, change the formatting because sometimes it reads in as a character and stuff like that. So those are other troubleshooting things. Okay, and then I'm just gonna copy this and then paste it here. Change that to Pdot, change it to Pdot here. So that's our phenotype file. If you open that up, you can see that first column is IDs. So that's just the sample number. This is a little more description of the sample. We have age, sex, COVID status, ICU status, and then a bunch of other things. Now I know that like there can be a lot of like different columns and measurements. So to quickly see the names of the columns, you just type in names, Pdot. There you go. These are all of the columns that you have, and these are all of the measurements. And yeah, so these are the ones. And so from there, you can very easily, like I said, you can view it as that, or you can do it as, so this is the table package is also used for viewing data and like working with data. I prefer to use them as data frames, but it's, a user's choice. So if I do that, it just shows it to me like that. There are several different methods that you can do it. You can use, okay, so then what I'm gonna use here now is something called as a pipe. So if you press Control or Command plus Shift plus M, this little symbol is gonna come up here. That's called a pipe. In the tidyverse universe or the tidyverse language, this is the command that you use when you want to perform multiple operations in one go. So what this is specifying is that I'm using the Pdot file or the Pdot data frame. On that data frame, I want to, let's just do the same thing as .tibble. Okay, it'll give you the same exact thing. So you'll see how this pipe comes into operation a little bit later on. So then you can also do like, there's another one called cable that displays it like that. So these are things, these are very optional things. You can go around, mess around with it and see which one you like yourself. What we're now going to do is, like I said, in our expression data frame, we have this one column which has all the names of the genes. However, when doing analyses, you kind of need to convert it into row names because some of the functions only want your numerics and they don't want any character formatting. And if it's present, the code or the function will not run. So what I'm going to do is for the Pdot data frame, I'm going to perform the function. So I want the output as Pdot, but I'm also performing it on the Pdot data frame itself. The pipe, Control or Command Shift M, enter, and then this command, column to row names. Now I want to name just the the name that we have for our column over here is gene. So you have to be very specific with that. So var means variable, gene. If you do that and you open up Pdot again from here, you'll see that now these are our row names. You also want to make sure that the sequence of the rows matches the sequence of the columns over here, because that's important, because a lot of the commands involve like using these two data frames to perform operations. And just to make sure that that works, we are going to do type in all call names of Pdot, and then just here is equal to Pdot $. Now what's its ID? So the name of the column is ID. That's what we're going to do. Let's see what comes up. True, that's perfect. So sometimes it might come up as false. That means that they're not in the same order, and then you would have to reformat that. Okay, and then now, let's see. Okay, so we'll now go into a little bit over here. I'm just gonna, oh, it's already here. Just check. Okay, we're gonna go into the Pdot Exploration, which means that we're just gonna look at a little bit of things over here visually, because that's always much helpful. Okay, any questions so far? Feel free to speak up. Okay, that seems great. So now we already have this little bit over here. Just let's leave that and we'll start over here. Okay, we already looked at all of the column names, right? The Pdot. Oh. Okay, these are all of the measurements that we have. Now what I'm gonna do is within Pdot, using the pipe operation, I am going to count the number of COVID patients that we have by the sex of that patient. So what I'm going to do, like I mentioned before, I'm specifically using the dplyr package, so colon for after dplyr, and then the count function. And then I'm going to put in COVID, because that's how COVID is over here. You have to specify the exact name. And then I'm going to put in sex. So if I do that, this is what we get. So this is just like, these kinds of, this is called as exploratory data analysis, or EDA. These things are very important to do before you even begin to analyze your data, because you need to know how is your data spread, like what are the characteristics of your data. Now, we can see that there is one unknown sample, which isn't, it's not great that there's an unknown, but also it's good because it's just one. So if we do need to, we can eliminate that if sex is something that we discover is like, a leading cause of variation. We can see that females who have COVID are 38 in number. Males who have COVID are 62 in number. Okay, and then we have females which don't have COVID as 13, males who don't have COVID as 12. If we just remove COVID, and let's just say, is it split well by sex? Because that is something that is a consideration that a lot of people forget, including sex. And in some cases, gender is very important, because that can inform you about, like, your project. Because let's talk about, like, for example, cardiovascular disease. So it's more predisposed to men is the general consensus, which is true. But did you know that after menopause, women are actually more likely to get heart disease? So these kinds of checks are very important before you even go into your data. Let's see. So 7451, that's okay, given that it's only a data set of 126 sample. It's pretty okay split. Okay, and then let's, you can just change this. Let's say COVID by ICU status. Again, so there are people who have COVID and are not in the ICU are about 50, but who have COVID and are also in the ICU of 50. So that's quite evenly split. There's also people who don't have COVID, but are in the ICU, which would be our controls because they're COVID negative. But then there's definitely something that needs to be accounted for or controlled when you're dealing with your data later on, because they do have a yes in the ICU column. Okay, is this kind of making sense to everyone? Just a thumbs up is fine. Okay, perfect. Okay, so who over here has no experience with R whatsoever? Okay, looks like everyone does. That's great. Okay, so then what I'm going to do is, this is a little bit of an assignment. Now we can see that if we go, like, I've built this workshop to be as interactive as possible because I want the audience to get out as much as possible. It's not just like you run this and you get this, okay? So if you go into Pdot and you look at your age column, this is present as a numeric, but a continuous variable. Now, if you wanted to check, does COVID actually have a implication with age or any sort of correlation, you would be able to do that. But if you do this, if you just put in age over here, you're going to get a bunch of things, like, because their age is a continuous number, right? So we're not actually going to be able to see if within certain, are younger people getting affected by contracting COVID more, or are the older age group doing that. So in this case, you would have to convert that into a categorical variable, which means that you want to place it into different brackets so that it's easier to compare. So if you worked with R, I would like you to make a new variable called as age category, age cat, and transform age into a categorical variable and check the spread of age versus within the patients as well as with COVID. Okay, so I'll give about 5 minutes over here. Don't worry, we're gonna go through this after. In the meantime, any general discussion, please feel free.
Nikita, can you maybe try and increase the font size?
Oh, yes, of course. Is this better?
Yeah, that's great. Thank you.
So this is like exploration on the what do you call it, metadata or the data, but it's not about the gene count, right? It's just
no, it's not.
Around,
This is about the characteristics of our data, but you have to remember, because we're coming from a biological perspective, right? Like, it's very important for us to know what the data is actually representing, not just the numerical value.
Yeah, maybe just to add to that. So if you're designing your own experiments, ideally you would think about all these variables beforehand, before you actually collect samples and send them for sequencing, to make sure that you'll end up with evenly distributed samples. But especially when you download samples, like already published data sets, it's important to make sure that you know what you're looking at and you're aware of the biases that are in the data set.
Exactly, yes. Okay, I'm going to start writing the code here. Okay, age category, right? So what we're gonna do is, like I did before for our Pdot data frame, where did it go? Yeah, column this little bit here. I'm gonna modify the pre-existing Pdot data frame, okay? So that, adding the pipe, and then one tidyverse function is called as mutate, which allows you to add new columns. I'm just gonna put in age cat, and then because we want to divide that up into different age categories, it's just depending on what kind of category you want. I didn't specify that, so it could have been just 0 to 50, 50 to 100 years of age, but I'm going to do it into 4 different ones. So because I want to specify it into 4 different categories, I am going to use the option case when. Okay, and then age. So I am gonna, I'm going to specify depending that if age, our variable age, is less than 20, then I'm going to code it as below 20. Then if our age is between 21 to 40, because we'll up until or like less than or equal to, if it's between 21 and 40, then I want to code it as 21 to 40. I'll copy that same bit here. Between 41 to 60, I'll put it as 41 to 60. And then if age is greater than or equal to 60, I want to code it as above 60. Okay, if we run that, I'll just copy paste this code in CodeShare so it's easier. Oh, okay, perfect.
Yeah, I already
there. Thank you, Christina. Yeah, and if I run that and you go to Pdot, you have to scroll all the way and you'll see, yay, we have an age category column. Perfect. And it's coded everything. So if we do that, it's from 21 to 40. So it looks like, okay, we have one below 20. That's it. And then we're going to do the same bit over here. So we'll do, actually, let's just check out what the class is of this. Okay, it's a character. What we're going to do is actually convert it into a factor. A factor means that it's going to group all of the same variables together. So instead of having all of these, we're just going to have 4 main categories because that's what we want to see, right? Otherwise, it might just be like every single data point that we see in the 21 to 40 just will be a separate measurement, and we won't get an idea of how it actually looks. So I am going to convert that. So Pdot$H as a factor. Just do that. Again, run this. Okay, it's a factor with 4 levels. Perfect. Now, we'll just do this. We'll just add cap. Okay, and this is what we get. And this gives us a better visual of how, if age is actually related to COVID or not. So let's see, for yes, we'll look at all of the yeses. 20 below 20, the person does have COVID. Okay. 21 to 40, we have about 14. 41 to 60, we have 29. Above 60, we have 56 patients having COVID. So it's kind of given us an indication that, okay, you know, the number of people who are having COVID might just be that they have an increased age. It just might be because the number of patients enrolled might have been at a, you know, higher age group. You have to also remember that there might have just been bias, or also we know that COVID affects older people, right? So that's just giving us a little bit of an indication of what our data is looking like. You can very easily just convert it into like a bar plot. And for that, I will write this code over here. I'll just copy paste it. So if you just copy that and paste it here, just change this to HCAT. And if you run that, so I'm going to explain this. So now we're using the ggplot2 package, which is used to plot and make all the graphs. Our x-axis is age category. Our fill is also age category. So each category is going to get a different color. We're going to use a bar graph, and then just set the statistic to count, meaning it's just going to measure the number of samples that we have. And then we're going to divide the graph by COVID status. And if you run that, this is what you'll get. So we can see that COVID, yes and no, and that the above 60s have, like, basically everything that we saw over here in this command just represented like graphically. There's like tons of options that you can use to modify this graph and make it better. Over here, we can see that below 20 is, you know, the last option. We can have it over here as the first one. But if you want to go through all of those extra things, just look at the part 1 file .md that's on GitHub, and then you'll be able to do this yourself. I won't go into that right now because I want to make sure that we get through all of these things. Okay, so then let's see, we will move on. Actually, before that, let's just look at the different protein measurements that we have. So I've just added this code over here. So if you just run this, you'll get all of the proteins with the COVID status in a separate data frame. And what we're going to do now is called as data wrangling. So we're going to convert this data frame into a bit more of a different format so that it's easily readable by the ggplot2 package that is required to output this. So what I'm going to do is proteins. Again, I'm just going to do it on the same data frame. And we're going to use a command called as pivot longer. We're going to use columns. Since what I want to do is convert all of these 1, 2, 3, 4, 5 columns, and not this column, I have to specify it either by name or by the index. So index meaning column 2 to index column 6. You'll see what happens with this code basically. So columns, you can say 2 to 6, or you can also say column minus COVID. And then I'm going to put the names to, which means that what is the column name that I want all of these column names to go into. I'm just going to put in measurement, and then values. So all the actual measurements that we have are going to go into, let's see, I'll put into, actually no, names to protein and values to measurement. Okay, run this and go into proteins again. This is what you get. So what it's done, this command has made a wider data frame into a longer data frame. That's why we use pivot longer. And proteins, I've put all of the names in this column called protein, all the measure, like counts in the measurement one. And then I'm just going to make a, actually I'll paste this code as well. So all these graphing commands, these are easy to find. That's why I'm just copy pasting them. It's not like anything specifically, and I'll like run through the code and what it means. So I'll just put it here. So I'll label this as protein plot. And then if you paste that here, what I'm doing again is taking the proteins data frame, ggplot, x-axis is COVID, y is measurement, fill is by COVID status. And I'm going to use a box plot. So that's why geom box plot. I can, I'll show you this option later. And then scale, fill, manual, basic, okay. Actually, let's do it like that. And then facet wrap with protein. And I won't put this in. So it'll show you what that means. and then the team minimal option. So over here, we saw that this is how it looked, right? So I'm just formatting a little bit. To run that, this is what you're gonna get. Now, the plot has automatically input the y-axis as being the same for each single measurement. In that case, we can't really see anything below, like we can only really see for ferritin and fibrinogen because they have higher measurements. And in that case, that's where the scales free comes in. So scales allows you, allows the y-axis of each plot to have variable limits. You can put in as actually free y as well to be a bit more specific. And you know what? I want to change the colors. So I'm going to put in scale, fill manual, values, and you just specify. So I want orange for COVID and gray for non-COVID. Just run that again. See, and it's much easier. So these are just things, you know, you can play along with. And this gives you an idea about, okay, it looks like CRP and CRP, fibrinogen, ferritin are lower levels in COVID patients, as well as lactate is a bit higher. So that just, you know, kind of gives you an idea about, okay, how are the characteristics spread? And then if you add this, this just shows you all the individual data points. So the shape is like, I'm specifying the circle shape. I want the circle to be gray. I want it to be a little bit transparent, alpha, and I want it to have a total width of the spread of the individual data points as width 0.2. For on that, okay, it doesn't give like the best because I've put in as gray, but you want to be able to see everything. So you can just increase this. So these are all the data points. If you put in, let's say red, it'll just show you the total spread of your data, which is good because sometimes you want to see the outliers, right? And then it's a very busy plot. So a box plot usually doesn't contain these. It's a violin plot that will have all of these data points, but I'm just showing you a couple of options. So that's kind of getting an idea about, you know, the types of proteins and stuff like that. Now, I'm going to pause here because now we're moving on into our expression files. Any questions up until now? So this is a little part which is called exploratory data analysis that is done. You can, if you click on this arrow here, it just pulls up everything. So this is the exploratory data analysis of our characteristics. Now we're going to look at our expression files. So we did start off that we had a couple of genes. Let's just see how many we have again. Okay, this I'm just gonna, we are gonna use this, but later. Okay, so we have 19,472 genes. And because this is just RNA-Seq, right? It doesn't contain, it might contain some microRNAs and some long non-coding RNAs, but these are basically gene names, like, no, actually present as these are present as gene names, which is great. Sometimes a single gene will have 2 or more transcripts, but that is something that we're not going to be dealing with right now. Okay, and then we're going to load in a package called as reshape 2. Now it's actually the same command as the pivot longer one that we used over here, but it's an old one, but I mean, I just wanted to introduce this one. So what we're going to do is we're going to convert this data frame into just 2 columns, one with the gene and one with the counts. And the way to do that is going to be, we're going to call it, so the function is called as melt. So I'm just going to name the new data frame as emelt, and I'm going to put in melt and then eat that. Actually, I think 3. No, 2. Okay, so what this has given us is that it's taken out all of the row names that we have, but it's just for each single sample, it's given us the value for each single gene. And I'll show you why I did it this way. I'm just going to rename the column. So call names is EMelt. Columns 1, 2 are going to be sample, and then the next one I'll expression to do that. Sample, expression. And now to see the spread of our actual expression data, I'm just going to run this. So this is, again, expression. I'm going to convert it into log2. So whenever you're plotting anything, it is always a good idea to convert it into log2, because when you have raw counts, there are going to be some which are 0, some of which are going to be, I don't know, 50,000, right? If we go over here, EMelt, and we look at the highest one, this is the highest recount that we have. So if you have a scale ranging from 0 to this, we're not going to be able to actually see a lot of the data that's there. So when plotting, a general rule of thumb is just convert everything to log2. So x-axis is going to be log2 expression. Color, I want it to be by the column sample. The fill is also going to be by sample, like sample as in I mean this column. I'm going to apply the genome density function. So a density plot is a smoothened version of a histogram. So where a histogram has bins that you can see, a density plot is just a like I said, a smoother version. So it helps when you have a lot of data. I'm going to set the transparency as 0.1. I'm going to use the theme minimal. So it changes the theme. It's a white background instead of gray. I'm going to put the legend position as none because actually, if I don't put this, let's see what happens. And then the labs, which means the labels. So x-axis is log2 reads per million. So all of these reads that we have are in reads per million. We have density as the y-axis. Title, I'm going to put it as sample distribution density plot, and then subtitle as raw counts. Now, whenever you're producing any sort of plots in any of your analysis, it's always a good step to make sure that you label it. There will be several plots that you'll be making, several ones that you'll be comparing to. And as long as you have labels on them, you won't get confused. Because trust me, you will when you have like tons of plots and you don't know what they're describing. And it's like a bunch of the same plots. If you do that, it might take a little bit of time because it's a lot of data. This is what we get. If you go to our console, that is basically the warning that our output. It says 544,000 rows were removed because they contain non-finite values. Now, that means that if you have a count of 0 and you convert it to log, you're going to get an infinity value. So in order for us to plot everything that we see, we have to convert that 0 into a numerical value. And this is something that is commonly done. So instead of applying a log2 transformation, we will apply I'll just paste this. We will apply a log2 plus 1 transformation. Now, what that means is basically that all of our 0 counts will have a count of 1 additionally added to that. It's not just the 0, all of them. So we're just shifting all of the counts by 1 in order to plot our data. It's not changing the spread of the data. So if I put that here, so again, the same data frame, mutate, adding a new column called log x1, which is log2 of the expression column plus 1. You go into email tier and you get log x plus 1. So if you have a 0, okay, that didn't work. It usually does. Okay, let's see. If I plot this again. Oh, and then another thing that I described is, we just add that here, log 2. Okay, and you do that. I think it's something with the way that it's coded. It doesn't usually give that. Yeah, so we got no warning, right? So that means that it's working. So everything's been shifted by 1. Now, what I was talking about is this theme position none. We can see that all of the samples have been given a color. That's 126 samples. We kind of don't need to see that right now because we're only looking at the spread. So I'm going to set legend position as none. So all of this is going to be removed. And this has to come below the theme option, because if you put it above, it doesn't work. To run that again. And this is what we see. Okay, so just looking from this, I'm going to open this up to the audience. Is anyone brave enough to just tell me what they see over here, or any conclusions you can draw from this? Okay.
Is it like normal distribution, but also left skewed? So
yes, exactly. So what it's showing us is that actually there's a lot of our our samples have a lot of genes or sequences that have counts which are basically 0, right? And even though it's like shifted by 1, it can show, it's showing you this. And that's not like, when we want to process it further, we don't want this, because a count of 0 is not informing us of like it is informative, but if it's 0 across all samples, right, then it's not informing us of anything. So what we want to do now, we're going to move on to something called as okay, we'll come to filtering a little bit later, but what we're going to do is now plot all of this as a heat map. And we'll just actually we'll make a object of this, just save it so we can use it later. Now what we're going to do is that we saw how the spread looks, right? We'll look at it, how it looks after filtering and normalization. But we also now want to kind of look at the correlation between our samples, like how are, let's say, the expression of genes of COVID patients correlating with those of non-COVID patients. So for that, we're going to do something called as correlation. And for that, we're going to make a new object called a samp core, which means sample correlation, and just this core. And for that, we're just going to put in Pdot. Now, you have to make sure that all of your gene names are as row names, otherwise this won't work. It has to be a numeric data frame. And the default correlation over here is Pearson, but you can also use Spearman if you want. You don't need to specify it because it's default, but that's up to you according to what your data is and what you're looking for. Okay, SAMP core. Oh, it's taking a little bit of time. Yeah, because we have so much data. Yeah, I mean, usually it doesn't take this long. Okay, perfect, it's done. So we have SAMCO here, it's a matrix. And this is giving you the correlation of one sample against the other sample. And I want to, you know, add some colors just so that it's easier to visualize. Because if I don't add the colors, this is how it's gonna look. I think you have this already. So, you know, if I just take this out here, you don't have to, I'm just showing you how it looks. So what we've done here is this is the code for the heat map. So we're using the pHeatMap function. It's the most basic one. There's another package called as complex heat map. I think, did we load that? Actually, yeah. Complex heat map allows you to play around with it a bit more. But anyway, for the purpose of just viewing our data, I'm just going to use pHeatMap. The clustering distance for each of the columns or samples is going to be the Euclidean distance. Again, this is up to you. What kind of statistics you want to infer? This is what I found works best for me. Clustering method is complete because the complete method of, there's I think 4 available, deals best with outliers, which we might have in our data. And then I'm going to put cluster rows as true, meaning that I want to see how do our samples cluster? How close are they to each other? Don't show column names, don't show row names, because that just, I mean, you can when it comes to genes and stuff, but over here, I'm just setting it as false. Set it as true. Annotation of the rows, I'm going to put in as, I want to look at COVID and sex. And then columns is also going to be COVID and sex, because we're doing sample comparison, not sample gene. And then just the name is going to be sample correlation. So if I run that, oh, what's this? Oh, I didn't. This should work. Let's see. Maybe I didn't.
Yeah, I can't remember, but like in your original script, you transformed you added the row names for your phenotype matrix. I can't remember if you did this already.
Yes, that's a very good point. Yes, I forgot that. So that is also very important. This is how we go wrong. So as I mentioned, we need to have gene names as row names, but we also, for our Pdot data frame, we also need to have the row names as the sample name, so that the column names of our expression data file match the row names of our phenotype file. So that's, I'm just gonna convert that here. Pdot row names is equal to ID. Okay, yes, that's a very important step. I mentioned it before and I forgot as well. Okay, so then I'm just gonna I'll keep this one. It's the same exact code, but just without the annotation color. So you should have had this one as originally. So if I run this is what we get. And then we can see that there isn't really any clustering by COVID or by sex. Everything seems to be spread across, but it's also difficult to see because the colors are not great. So I'm gonna add this, and I'm just gonna name it as annotation columns. I'm going to give COVID, if it's yes, a gray color, no, orange, and for sex as well. And then if I run this is what we get. It's much easier to see. But then again, we can't really see anything by sex or COVID. What if we added age category? Let's see. So if I add age cat, nothing really. Again, I don't have colors for this one, but it doesn't seem to be clustering. So that just shows us that it doesn't seem like COVID or sex or age category are variables that seem to be driving the gene expression that we're seeing. It might just be some other inherent characteristics. We also don't know the populations or the ancestries that these patients come from, right? So each population has its own different spread of expression and like different genotypes. So that is also something to account for. And that's basically, so our data is telling us it doesn't seem that having COVID, if that's our question, is driving the expression or is separating our samples by the expression. Okay, and then now we're going to move on to the more quality control that we have. And okay, for this one, we're going to apply like a couple of steps. So this is just for the plots, but we're, as I mentioned, like we're going to perform a different, like few different filtering criteria. First one being, I just want to get rid of all of the genes that have an expression count of 0 across all samples. Like I said, that's not informative. So I'm only gonna keep all the sequences with the read count of more than 0, or basically more than or equal to 1 in all samples. So I think I'll just paste this. Just give me a sec. Okay, yes, for quality control. So this is for reads of greater than or equal to 1. Okay, so that's what I want to keep. And if I paste that here, so I'm just going to go with E for expression, fill for filtered 1. And if I just run that, so what I'm going to do now, we know that our, let's get rid of that. Our gene names are as row names. So when performing this function, like performing a lot of functions, R gets rid of the row names. We don't want that. We want to make sure that they're kept. So what we're going to do is we're going to, within using the eFill, like making the new object, using the e.data frame, we're going to convert the row names back to a column called as gene. Then we're going to filter at all of the columns besides the gene column, because that's our gene column, right? It's not numeric. And then we're going to keep only the rows that contain counts of 0, 1 or more. And then we're going to convert the columns back into, like the gene names back into a column. And if you run that, this is what you get. Okay, so in this case, I removed all of the genes that contained a 0 across all the samples. Now for here, you can see that, okay, this is 0, but there is a count of 1. So that's why the A1CF gene was kept. You can very well make sure to remove. So, and then we're just gonna plot this as G-FIL1. You can actually see the number that was retained. Okay, so that's 18,000. So our original was 19,000 something something. So that removed a bunch of genes. Now, we can actually just convert this into, just to plot it again as we did for the density plot. Just gonna melt it. No, 1. And then again, now we know that there are quite a few that have the 0 count. So we're gonna run into the log converting problem again. So what I'll do is just go over here, copy this, paste it here. After like you've written most of your code, it's basically just copy pasting. So log x1 log2 expression. So actually over here, we have to melt it first. And our column is, no, sorry. Our column is called a value. I haven't changed the column name, so just value +1. We get this, and then it's basically the same code that we ran from earlier, just using this. So we'll just store it as an object. We're now going to keep only sequences or genes with a total expression of 1 in each single sample. So for that, the code is a little different. It'll take a little bit of time to write it, so I'm just copy pasting. Okay, and then what we see is, again, using the EDAT data frame, converting the gene to a, like all the genes to a column, filtering at -gene, but instead of any variables, which are just keeping the sequences with, removing all the genes with 0 across, we're only going to keep sequences which have a count of 1 or more. And then just column to row names again. We're going to do this again here. So we had 18,000 before. No, sorry. Yeah, right. So 1 was 18,000. Now we have 11,000. So that removed quite a lot. Again, all of these filtering steps depend on what criteria you want. Do you want to look at a range of expression, or do you want to look at only genes that are definitely expressed or highly expressed? That I'm just going through the different criteria that you can use. So that's 11,000. That's a lot of genes that have been taken out. So then I'll put this as melt fill 2, and then this here. Given that we don't have any zeros, we don't have to deal with the log problem. So I can just make this directly. And this is something, again, if you're going into more of a discovery phase and you're wanting to see, like a full profile of the expression that you're getting. Some genes that might be expressed in some samples might not be, like a total count of 0 as well. So in that case, then what I'm going to do is I'm only going to keep sequences with a read count of more than 1 in 30% of samples. And in that case, then we have to like calculate what 30% of our total 126 samples is. So what I'm going to do here, and this is G. There was another. So for this, basically, if you go in the main file, you'll see that you can do it by, I've done it RPM of more than or equal to 1. You can do it by 2. I'm just not including that. It's just changing this 1 to a 2 over here. So that's why it was G full 4 instead of 3, but I'm just going to change it to 3. And then what I'm going to do is, if you want to like look at, let's say, the total, what is 30% of 126, right? So that's going to be 30 into 126 divided by 100. So this is 37.8. So you know what, we'll take like 38 samples. So for that, what I'm going to do is, okay, this is a bit more. So copy paste all of that with the comments that I have in the code share. All of this is again found in the main document. So what we're going to do is use these steps here. So let's explain them now. So what we're going to do is efill 3 is going to be taken from EDAT, and it's a bunch of operations applying now. If we're going to filter across each of the rows, filter is used for rows. We're going to take all the columns which are numeric. So that leaves our column, which is like the gene column. We are going to sum them up, and we want all of them to have at least a read count of 1. And if the total sum is more than 38, so we have 1, 1, 1, 1, 1 across, and the total sum for that gene is more than or equal to 38, we're going to include those. If you run that, you'll get efill3. So basically all of these, we can see there's quite a few zero counts, but if they all got to more than 38, then we've kept those. And now this is something which is a bit more like, you can read the description over here. I won't go through that because I want to get through everything. So if you just run that, it's going to show you the total number of like which column has how many row sum counts. Just run that. Okay. Oh, efill3. And then you can read about this in your own time. So yeah, so that's 3,000 sequences removed if we go according to if it's present in 30% of the samples at a minimum read count of 1. And then I'm just gonna do this as an object, and then that over there. Now, if you want to just look at all of those together, what I'm going to do, gg arrange from the ggpubr package, I'm going to put in gfill1, gfill2, and gfill3. If you run that, you get over here we would have to convert it again because we won't have zeros. This is what we get. So if you just enlarge, you can click on this in your own console, because if I do that, you won't be able to see. But basically, this is showing you the spread by the different filtering methods that we applied. Now this one, so the first one contains, oh, this is wrong, contains all the samples that had at least 1 count across, like in all of the samples. So we do have a lot of zero counts here. Here we got rid of all of the sequences with 0 counts. So that looks a bit better. Over here, we took out the ones which had at least, which were kind of one in at least 38 samples. So we still have some counts of 0. So this just gives you an idea about the different, like how you filter your data is going to be very important in the downstream analysis that you use, because this is basically feeding into the main genes that you're selecting for. Again, this is more of a informed decision that you have to make. And it totally depends upon your project and what you're wanting to get out of it. Like I said, is it just sequences that you want high expression in, or do you want to do more of like profiling? Christina, do you want to add anything to this bit?
No, I think that's great.
Okay. Okay, so now we're going to move on to something called as normalization. Normalization, if you work with a little bit of data, means that you're trying to get all of your expression counts into a like scaling them so that they're comparable across each other. Now, okay, I'm just gonna paste this because I'll explain it step by step, but sometimes co-chair takes a little bit of time to load. Okay, so basically, normalization is what you perform when you want to scale all of your data. So sometimes there are going to be samples that have an extremely high expression, and some samples are going to have low expression. Now, how are you going to compare these? Just because maybe, you know, a sample for one patient was taken after dinner, one was taken before dinner. That usually doesn't happen, but that's just an example. So you want to be able to compare the expression and not just say that this sample is having higher expression because of some fluke value. So normalization, you know, kind of tidies up your expression a little bit more. And in this case, I think, yeah, just to go as an example, we're going to select the filter 2 data frame that contains counts of more than one, at least one or more than one in all of the genes that we see. And the normalization method that I'm performing is called as RLE, or relative log expression normalization. There are a bunch that exist. There's TMM normalization, trimmed value of M means, or there is just direct, you know, counts per million normalization, which we will be applying. But I found that RLE, after like my extensive research, that performs better because not only does it take into account that there is going to be within sample difference in expression, so because of conditions, you know, just in that one sample, but there's also going to be between sample expression differences, and RLE specifically works best for biological samples, at least in my opinion. TMM is great as well, but then if you read like papers which compare TMM versus RLE, some find that it's better, some find that it's the same. So again, you have to make that decision according to what normalization technique you want to use. But this step is extremely important because otherwise what you're dealing with is called raw counts, and usually that's just not like what you do. Normalization is a very essential technique that needs to be applied. And if you're all going to be dealing with your own data, I would highly encourage you to go and, you know, read a few papers, check a few tutorials to do with normalization and what it entails. Okay, so we're going to use the eFIL2 dataset, and then I'm going to put all of the genes that are present after our filtering in a separate object called as genes. So I'm just gonna take the row names because that's how we have it. So if I go there and I click on genes, we get all of the genes that we wanted. This is the command that we're running. So norm, I'm gonna name the object norm. We're gonna convert it into a DGE list type object, which is essential for the package that we're using, which is HR. HR only takes in DGE list type objects. So we're going to put our counts, our expression counts, as the eFIL2 dataset, samples as the Pdot dataset, genes as genes, and group is, so you can include this, you don't have to include it, but we're just saying that, okay, our variable of choice is COVID, so that I'm just going to put that in. And if it normalizes by COVID, let's see if there are any differences. Just run that. I'm going to name our normalized data frame as eNorm. Now, this is where the actual normalization command works. So the command is called calc norm factors. We're going to apply it to our norm data frame, which is a DGE list type. A method is going to be RLE. Over here, you can specify which method you want. Okay. And then we're going to scale. So RLE performed between sample normalization. Now, once that's done, we're going to do within sample normalization. So counts per million, and then as data frame. So if you look at that, this is what we get. So this is our original eFill 3. This is our original data frame. This is going to be our filtered data frame, and this is our normalized data frame. And from now on, anything that we do is going to be on the normalized data frame. And just to see how different this looks, I'm going to again melt it, melt norm, and I can save this as M1. And then again, GG arrange, I'll put in eFill 2, actually, I'll put in all emelt, eFill 2, and M1. Actually, no, this won't be it. Maybe I didn't specify. Oh, G1, sorry. So this is the progression of our data now from like raw counts. Okay, what is happening? GFill. Yes, this is a progression of our data from raw counts to filtered counts to normalized counts. Oh my. Oh, right, okay. This is not what we wanted. Need to change that. Melt norm is value. Ooh. No, this is actually.
I think it should be, yeah.
Yeah. Okay. Yeah. Okay, so you can always obviously like arrange this better, but this is what we see. This started with our raw count. We moved into our filtered data set, and this is our normalized data set. So this should not be log 2x plus 1. But that is what we see. And as you can see, you know, this is definitely cleaner than this one. So kudos to us. We've gotten to a good workable data set. Now over here is just a little bit of an exercise. Firstly, because I don't want you all to forget this, I want you to save that data frame first. So if you just run this enorm to enorm, so that'll mean like all our genes are here in a column so that we won't lose them when we save them as a file. And then just this little bit here. So write delimited, like the file will be a delimited file, enorm, which is a normalized data set, like the location of where we want to send it, save it. So file, here in data, and we want to save it as enorm.txt with the delimiter /t. Just run that, and you go to data, you have your enorm here. So if you don't see it, just refresh it. That should work. And over here, this is just a little quick, what do you call it, exercise. So what I want you to do is just run all of this. And where you have your X, okay, I'll just run through this. So random sample. So we're going to select a random gene, and we want to see if like how that gene actually compares to when it is raw counts versus it is normalized counts. So we're actually not even EDAT. This should be eNorm. Okay, so random sample. So if you go into random sample, you'll see you have the DPYD gene. Everyone will get a different gene. So, and then row names. So, okay, that sometimes works, but sometimes doesn't. Okay, so what we're gonna do is random sample. You're gonna copy this, whichever gene it is, and put it wherever I have the X. A better way to do that is actually if you select X wherever it is, you go into code in your RStudio, you go in rename and scope and scroll down to wherever it has backspace and then just paste it. And that's it, done. Okay, so if I run this, oh, it's already in gene. So we don't need actually row names to column. You can take that out. If you don't want to run something, you can just put a £sign before that. So R, okay. Random sample from eNorm. Arguments do not match. 126. Oh, 127. Why? Oh, okay. So because I ran it again, that's what has happened. But this should run. So if I just remove random sample, but you should be able to do this without any problem. Okay, I'm going to take another random sample. It's the FOS gene. And then it doesn't come up with row name. So FOS. Okay, it should work. Okay, I'm just going to take out random sample. So I'm just removing the first column. Actually, this is going to be, we're getting it both from the normalized A-frame. So this is going to be sampled from raw. So keep that. Okay, so it's not present. So, but basically what we want to do is once we get this, and I'll just show you how it looks like. So if you go here and you compare two of the whichever gene that you choose, you can see that for the raw, which is the TRIM62 gene for the raw, the values are spread all across. But then for the normalized one, they're all in a pretty good line, meaning like they've been scaled to actually show what the expression is in relation to other genes and within the same sample as well. So that leaves us with everything. Once we've saved our data frames, that's it. And that's the end of part 1. Any questions so far?
Do we have time for question, or you want to leave it?
I'm good to stay, but whoever, I guess, Lorenzo, are we okay to stay?
Yeah, we can stand for a couple more minutes. Yeah.
Okay, thank you so much. Great, Nikita. Could you please just briefly explain what happened in normalization? Whenever I do have gene expression normalization, it reminds me of using housekeeping or reference genes and dividing the expression, getting the normalization. But here I didn't see such a thing happening. So what was going on during normalization exactly?
So okay, so what normalization basically does is when you do that, it gives a positive or a negative integer or like numeric value to each single gene that you have, each single sample, each single gene. And that numeric value is either subtracted or added from the raw counts. So this is all going like behind the frame. You can go and like check out the actual object. It'll show you the actual, you know, the numeric value that is either added or subtracted. So the calc norm factors actually does that. And once like, and it does it according to the full data set that we have. So then those numeric values, like I said, are added or subtracted from the raw counts, and that's how you get your normalized data frame.
So basically it has nothing to do with the biological information that it has, for example, the one that you mentioned before dinner, after dinner, or any sort of treatment. It's just a kind of mathematical calculation, am I right?
Exactly, yes. This is all just to do with the counts that we see. It's a, what do you call it? Just, yeah, scaling it down. And then just the RLE, like I mentioned, it does, however, like have an inbuilt function, or like the reasoning behind the RLE normalization, that it knows that it's working with biological data. So it, like I said, it doesn't do it just within each sample, it also does it between sample. That's the difference. Because if you just use like interquartile or quantile normalization, that works just on like, you know, just a set of numbers. It doesn't know that there are any trends, which we know that are in biological samples. That's why I choose that one.
Okay, perfect. Thank you so much.
Yeah.
Will you cover BCCHR in your next session?
Yes,
I'm asking because, like, I think like you don't have to normalize in BCCHR, right? You have to, as an input, it's just the raw counts. Am I right?
Yes, there are 2 ways to go about that. I myself have been confused about this, but it basically says that you can either go with raw counts, is what the literature, like the documentation says. But then if you do that, I think I can't remember exactly, but I can send that to you if you want. But there are people who use raw or who use normalized.
For these, like maybe you'll cover, but for every like pipeline, do you have to remove zeros? I know you said like it depends on your question if you're interested in that expression or not, but these filtering steps that you did earlier, you really have to do that for every like if I'm feeding the DSIC to the raw count, do I have to remove all the zeros and add 1 and things like that? No.
I don't think so. No, like I think the package takes care of that, because it's only the removing of the zeros is it automatically, I'm pretty sure, removes all the samples, all the genes that have zeros in them, because like, you know, we're not getting anything out of it. And yeah, I think it does it behind. It's just like all of these methods, I prefer to do them myself, so I know exactly what's going on, right? And then I apply exactly the method that I want. And I don't leave it up to, like, if I can do it myself, I'll do it and then feed it into whichever package I want.
Yeah, thank you. Are you guys sharing the recording in the email or something?
Yeah, you can get in touch with the GrasPods email address. I'll share it here in a second.
What's their email? Is the GrasPods?
Yeah, just give me one second. I'll share it.
Sure.
Oh, Maren, I just saw your message. Yes, like as Lorenzo just mentioned, you will get the recording if you want it. I think I'll email Diana to tell her she can put it up on YouTube as well. So, you know, and then if you just follow the code that like the link that Christina has posted, you can find the full workflow of what I've done with even more detailed annotations in the part 1 and the part 2.md files.
Okay, awesome. Thank you. Are there any other questions?
Thank you.
Okay.
So Nikita, you mentioned there's a lot of events happening and workshops. You got the training. Sorry, I forgot the name of your
Training
group. So
yeah,
is there like a sign up sheet that you can? Oh, you provided a barcode. Sorry, I forgot. Yeah.
No, don't worry. If you do sign up to our mailing list, you'll get to know all of the things that we're gonna be putting on. But I can also ask GrasPods. I'm sure GrasPods can like distribute TOG information, right? So like if you wanna like sign up to any of that, just do it. Like, or if you just visit TOG like BCCHR/TOG, you can see what we have over there as well.
Okay, thank you, everyone.
Yeah, of course.
There's no further questions. Nikita, thank you so much for that. That was amazing.
Thank you so much, Christina, as well, for supporting me throughout.
Awesome.
Okay.
Both were great. And yeah, so we'll keep in touch with everyone for the recording, and then we'll also distribute the materials for part 2, hopefully the next couple of weeks.
Perfect. Awesome. Great.
Thanks, everybody.
See y'all.